Well, hello and welcome to everyone joining us for this X-Talks webinar. Today's talk is entitled Innovative Approaches in Metabolic Dysfunction-Associated Steatohepatitis Clinical Research. My name is Corey Stanton and I'll be your X-Talks host for this event. Today's presentation runs for approximately 60 minutes and the webinar includes an interactive Q&A session at the end. To make the most of this session, feel free to submit questions and comments for our speakers throughout the presentation and we'll try to attend to as many as possible. If you're joining us via GoToWebinar, you can use the questions chat box, which is located in the control panel on the right-hand side of your screen. And if you require any assistance, please don't hesitate to contact me by sending a message using this very same chat box. And if you're viewing this presentation from our LinkedIn Live event, you can submit your questions using the comments tab on LinkedIn. Please note that all audience members are in listen-only mode and that this event will be recorded and made available for streaming to those who registered on xtalks.com. At this point, I'd like to thank Medpace, who developed the content for this presentation. Medpace is a scientifically driven, global, full-service clinical contract research organization providing phase 1 through 4 clinical development services to the biotechnology, pharmaceutical, and medical device industries. Medpace's mission is to accelerate the global development of safe and effective medical therapeutics through its high science and disciplined operating approach that leverages local regulatory and deep therapeutic expertise across all major areas, including oncology, cardiology, metabolic disease, endocrinology, central nervous system, and antiviral and anti-infective. And now let's take a moment to meet our panelists for today's presentation. One of our speakers today is Dr. Sal Zavattino. Dr. Zavattino has over 30 years of experience in pharmaceutical and device clinical research. As a founding member of Medpace, he has held leadership roles in clinical operations, regulatory, biometrics, IT, and cardiovascular services. His focus is on integrating clinical, operational, and technical aspects for multinational studies across multiple therapeutic areas. Dr. Zavattino has extensive experience with imaging and biomarkers for drug and device safety and efficacy, including DXA, ultrasound, cardiac echo, MR, CT, photography, pathology, ECG, Holter, and biosensors. Also joining us today is Dr. Robert McGee. Dr. McGee is a board certified in both anatomic and clinical pathology and brings 25 years of combined experience in diagnostic and central laboratory anatomic pathology, histology to Medpace. He's an active committee member of the College of American Pathologists with assignments to the Histotechnology Committee, Digital and Computational Pathology Committee, and Reproductive Medicine Committee. We're also happy to welcome Dr. Nicola Owen. Dr. Owen is a board-certified gastroenterologist and hepatologist with 19 years of clinical experience, including 11+ years in specialist hepatology and gastroenterology. Prior to Medpace, she was a consultant hepatologist at King's College Hospital, leading the Advanced Chronic Liver Disease Service and serving as a principal investigator in clinical trials. She managed complex liver disease cases and received referrals from across the UK. And rounding out the panel today is Jensen Ruiz Garcia. Mr. Garcia started his clinical research career at Medpace in 2018. He has 9 years of experience in managing phase 1 through 3 trials, including a successful working relationship and collaboration with flagship sites and KOLs. He has demonstrated a passion for research, risk-based management approach, and leadership skills throughout his clinical career, where he has led multiple large global outcome trials. Mr. Garcia received a Master of Science in Molecular Biology of Infectious Disease from the London School of Hygiene and Tropical Medicine in London, UK. And now, without any further ado, I'd like to hand the mic over to Jensen to kick off the presentation. Jensen, the floor is yours.
Excellent. Thank you, Corey, for that introduction, and thank you everyone for listening today. Then we'll jump onto our agenda slide. So we're really just going to be discussing in terms of some of the key operational considerations we have in our NASH/MASH studies. Then we'll hand over to our team, as we've seen today, really looking at the histology, the anatomic pathology, the evolution in the read paradigms and that paradigm shift that we've seen across MASH studies. And then we'll go through a summary, really capping what we've gone through today, and hopefully have enough time to go through a number of your questions, and hopefully we can answer all of them later today. So with that, thanks everybody for listening, and we'll jump on to our next slide. So I'm going to start with the end points, so looking towards the end, and really NASH has seen quite a paradigm shift, even from the consensus and update now identified as MASH studies, really looking at it that cardiometabolic dysfunction-associated hepatitis. And really these trials do rely on that gold standard liver biopsy when they're assessing the histological features. So really looking at the level of stress that the liver cells have, and we're measuring that by determining the steatosis, the ballooning, the inflammation that that the patient populations will have, and that is critical not only for the eligibility and what's driving the patients into the trials, but also for our endpoints when we're looking at our long outcome studies. Now, the FDA and EMA guidance in terms of drug development for endpoints and MASH outcomes is very key, that we do require liver biopsy for disease progression, and it encompasses it as part of their composite endpoints that we have. So we're also looking for the progression of the disease, the improvement in the quality of life that the patients are having, but ultimately it is that biopsy that's driving that key point and those endpoints that we have and what they're looking for marketing authorization. Now, our agencies do recognize that with the biopsies, there are potential concerns, and they are really proposing the use of non-invasive tests. So the MRI-PDFF we have for the liver fat content, we're looking at various biomarkers as well. However, at this stage, these non-invasive methods are not sufficient to replace the biopsy because they're just insufficiently validated for our studies in MASH. So while we understand that MASH represents a growing major health concern and one that continues to grow, and depending on the projection you'll see, it is due to impact quite a large patient population, create quite a heavy burden. Studies involving MASH do include very invasive screening and endpoint procedures, and these will impact overall patient recruitment. So before we really look into the biopsy itself, we always have to think about the overall study design, really making sure that we're opening the study up and focusing on the patient centricity aspects of it. Not only do we want our patients to go through the door, feel part and welcome of the study itself, but retain them so that we can have that key biopsy and that output. Now, we understand that the biopsies come with a significant patient burden. They're invasive, they have high risk pathways, they're affecting the eligibility, and a lot of patients will be scared and concerned this, so that education and awareness, making the patients know that they're part of quite a significant journey, and there is a huge shift going on right now in MASH in terms of potential drug aspects as well for this population. And education, there's a high propensity of patients now who have high liver fat, they have all the other comorbidities and just are unaware of this. So all of this really does help us in terms of preserving the critical endpoints that we have in these study designs. And focusing on that patient centricity, just those things that we can focus on really is that patient education. So calling out that we do understand that around 2% of patients will report complications from a biopsy. So if they're naive to it and they do not understand the risks are minimal, but we have to promote that and just make sure that that key education to the participants is there and is constantly repeated, and they're aware of what it is that they're doing and what their scientific contributions are. We do find that when the participants are made aware of what is it that they're contributing to, the impact that they'll have to return for that critical endpoint biopsy is protected, and they're more involved in the whole study design. So really developing that strategy from that perspective supports them, and trying to again minimize the overall biopsy burden. What is the frequency of the biopsies that are being proposed in the clinical trial, and what are the risks that are being put to the patients? And again, trying to supplement with what the agencies are calling for in the proposals to look at those non-invasive tests, so we're not left behind, and always looking to see what other data we can get to really elucidate the pathways to combine the biopsy and all those other non-invasive tests. However, saying all of this, we do understand that the liver biopsy is critical in understanding the patient population from an eligibility perspective and an endpoint perspective. So with that said, we'll hand over to Dr. McGee, and we can go over to Rob for our next slides.
Thanks, Jensen. So thank you for setting the stage about the liver biopsies. So what I'd like to do for my set of slides here is talk about the histology lab, and from the point of the view of the pathologist, because what we do in the histology lab for the processing and handling of these liver biopsies does have an impact on the interpretations that are being provided by these key opinion leaders in the MASH space, in particular MASH pathologists. So for this talk, I'd like to just focus on a couple of lessons learned from the setup of MASH studies over the years. So I've got two topics I'd like to address here. The first is the standardization of the pre-analytical variables that are involved in the handling of these liver biopsies. And then secondly, I'll address the question of what are the best samples or preferred sample types for a MASH study. So whether you're talking about any kind of test in the laboratory, from the time the specimen is received until the report is generated, There are 3 phases of testing that can be approached or looked at in terms of handling and processing, analyzing, and looking at the results of those reports. Those 3 phases are the pre-analytical variables, and so that would include all procedures that occurred during the sample collection leading up to the actual sample analysis. The analytic phase, the central phase, is the actual measurement of that target, whatever that target may be. And then finally, there is the post-analytical phase, which is the collection of the results, formulating the pathology report, and sending the pathology report out to the physician. So with regards to the pre-analytical variables that are involved in the handling and processing of these liver biopsies, I've got a short list here, but these variables include the sampling method, which I've got a slide that will follow here in just a second, the handling, the fixation, and the fixation would involve the type of fixative, the volume of the fixative used, the time of fixation or the total formalin exposure time, and the temperature of the fixative agent. Could also include things like processing schedules, which would include the reagents, times, temperature, and instrumentation used for processing the biopsy. And then finally, microtomy, which would be the sectioning of the paraffin block to create the unstained tissue sections. If nothing else, what I'd like you to take away from this discussion here is that the preparation and standardization of the liver biopsy samples requires good communication from the site to those involved in preparing and handling the sample. This is a key element to ensure that the biopsy that the outside pathologists are going to get gives the subject the best opportunity to be included in the study if the histologic features of MASH are present. Now, what would that mean? Well, it means that the site needs to either do the biopsy themselves and be aware of what the requirements are for an adequate liver biopsy specimen, or it may mean that the investigator site just simply needs to call the outside imaging center. If an imaging center is going to be used, a third-party imaging center, that's going to do the biopsy and indicate this is a subject for a MASH study. These are the tissue requirements that we would like to have in order to give the best opportunity for the subject to be enrolled. So the question then becomes, what's the best or what's the preferred biopsy specimen for a MASH study? Now, there is a publication from 2022 where a group of gastroenterologists and MASH pathologists published this article on the best practices for the handling, processing, and what they felt were an adequate liver biopsy for a MASH clinical trial. So from that paper, the recommended biopsy size is to use a 16-gauge needle biopsy. Now, some places are hesitant or reluctant to use a 16-gauge needle biopsy, but if one can, the minimum length here should be 1.5 centimeters in length, and up to 2.5 centimeters would be ideal. And you can see down here on the bottom left panel is an illustration of what a 16-gauge needle looks like. If there is a hesitancy to use a 16-gauge needle, then the recommendation is to use an 18 to 22 gauge needle. But if one's going to do that, then you need to use or obtain 2 to 3 cores of tissue with a total tissue length of equal to or greater than 3 centimeters. And you have an example down here in the middle panel of an 18-gauge core, and there are 2 cores in it. So you can now compare the 16-gauge volume of tissue, if you will, to the 18-gauge with 2 cores. Now, what we've seen sometimes in the past is if there is no communication and that outside imaging center is just told to get a liver biopsy, sometimes we end up with these 21-gauge single core biopsies. And as you can see, and I've got an example here on the bottom right, if you look at just the amount of tissue that the pathologist has to evaluate, there's certainly a lot more tissue in that 16-gauge and 18-gauge with 2 cores compared to the 21-gauge. So what we're trying to do here is optimize, again, this biopsy to give that subject every opportunity to get into the protocol and into the study if possible. I will also say, sort of parenthetically, that with a 21 gauge, the chances or possibility of seeing those rare balloon hepatocytes does decrease. So again, getting an adequate biopsy here is paramount to give the subject the best opportunity to get into the study and has the histologic features of MASH. There are a couple of other variables that I'd like to address here. The first is the formalin type. 10% neutral buffered formalin is a pretty standard histology fixative. It's the workhorse of the fixatives we use in the histology lab. But there are some regions of the world, and most notably we've seen around the Asia Pac area, that do use a higher concentration of formalin, and that formalin could be buffered or unbuffered. We've seen examples of 16%, 20% buffered or unbuffered. Now the formalin type does have a bearing and can alter the staining characteristics of these liver biopsies. So having a standard buffered formalin, 10%, helps reduce that variability in the staining of these liver biopsies that are being sent to the pathologist. Now, I'd also like to address a second variable, which is the total formalin exposure time. The amount of time that the biopsy is exposed to the formalin can also alter the staining characteristics and parameters of these liver biopsies. So it would be best to standardize this exposure time to roughly 6 to 72 hours is preferred, and that includes the shipping time. So if the biopsy is going to be shipped, and it can be in formalin, and it can be received within 72 hours, we don't need to do anything. But if the biopsy is going to be in that formalin for more than 72 hours, the recommendation is to fix that liver biopsy overnight at the site and then transfer it to 70% ethanol the next morning and ship it the same day to the central laboratory. Now, the reason I mention these two other variables is because there is right now AI software and programs that are being developed to measure some of the features of these liver biopsies. And here I'm speaking directly about fibrosis, collagen, the type of collagen that's in the fibrous bands. There are AI programs that are being developed to actually do the NAS scoring of these liver biopsies. So if artificial intelligence is a thought or may be used in a clinical trial, standardizing these two variables will go a long way in terms of presenting a uniform product to an AI algorithm for its analysis. So what we're trying to do here really with the central lab, histology lab, and centralizing these pre-analytical variables we are trying to minimize the observer variability that we know exists out there between pathologists, and we're trying to minimize the turnaround time based on the processing of the tissue, as I mentioned, as one of the pre-analytical variables. What we're trying to do here is presenting to the outside readers a uniformly stained glass slide or a digital image of the glass slide on a platform that they can then use to score for entry into the study as well as their end of study biopsies. All right, so now let's turn our attention to the question that seems to come up quite a bit is what's the best sample type for a MASH study? So I've put these in rank order. The first or preferred sample would be an original uncut paraffin block. So this would be in the scenario where the biopsy is obtained and a local pathology lab simply processes the biopsy, puts it into the paraffin block, and sends the paraffin block to the central lab. Obviously, there's going to be a lot more tissue in that block for us to cut and stain because it has not been cut by the local lab. And sometimes the local labs get a little too aggressive in taking the sections or facing the blocks. And so they cut too far into the block, into the tissue, and don't give us the readers the best sections from that block. So the first preference is an original uncut paraffin block. Second choice would be the historic cut paraffin block from the site. Now, the downside to this is that the local pathology lab, again, may have been a little too aggressive in cutting that block, and there's little residual tissue left in the block, but there is tissue, but that's what's left. There can't put any more tissue back into the block. It is what it is. But this also brings a second point about communication. So if the subject undergoes the biopsy, the site can call the local pathology lab and also tell them that this is a subject for a MASH study. Please be careful, please be diligent, please take caution when cutting that block such that you retain as much tissue as possible in the block, because the block's going to go to a central histology lab and process there for more additional stains. So again, communication here is key in terms of preserving as much tissue as possible for the downstream testing that is going to occur in a MASH study. The third option here is if the local pathology lab is unable to release the paraffin block to the central lab, then it is permissible for the histology lab, local lab, to make unstained tissue sections from that historic paraffin block. And then finally, the last option here is the fresh wet tissue biopsy, which may sound kind of not intuitive, but it's been observed here that if we get one of those little small 21 single gauge, 22 gauge single core biopsies, those are really frail and friable, and they can break apart and become fragmented during shipping. So that renders the biopsy almost uninterpretable or unsatisfactory for the pathologist to score. So again, first preference is the original uncut paraffin block, then the historic block, unstained tissue sections, and then lastly, wet tissue biopsy. And with that, let me turn it now over to Dr. Zabatino, who can talk about that NAS score that I just alluded to in terms of providing a stain slide for the pathologist to read.
Thanks, Rob. So clearly we've now heard how not all slides are created equal and how important it is to create a set of images that are optimized for our central review. The first challenge that we have is who's going to review this, and this is how at Medpace we turn things over at this point to the Medpace Core Lab, who coordinates the review and the scoring of the slides for the purpose of eligibility as well as for the efficacy and safety endpoints that are going to be used in these clinical trials. From an imaging perspective, we're often faced with the challenge of how are we going to evaluate the images that we're looking at. And so thankfully, Dr. Kleiner and a team of other pathologists, 8 CRN and the 1 NIH pathologist in 2005, came together to try to create a semi-quantitative scoring system that we could use in our clinical trials or natural history studies to assess change over time for these things. It wasn't necessarily to create a scoring for rapidity of the disease progression, but really something that we could use to semi-quantitatively score and see where we are with it. So they came up with a scoring system. They came up with 50 cases, 32 adults and 18 pediatrics cases. They used H&E and trichrome stains that Rob has done a nice job of describing for us and how we prepare those. So what was the scoring system that they ultimately ended up with? And if we move to the next slide, we can see something that many of us are very familiar with and have been using for the last 20 or so years. A scoring system that kind of broke things out into 3 basic areas referred to as the NAS scoring. Steatosis, kind of very simple in concept. Less than 5% of the slide showing steatosis was a 0. About a third gave you a score of a 1. A third to 2/3 a score of 2. More than 2/3 you got a score of 3. And so that seemed pretty straightforward. As we moved to lobular inflammation, they looked at the number of foci that were being seen in that field of view, and based on that, you got a score of from 0 to 3. And then once we get to ballooning, and we'll hear a little bit more about the challenges in ballooning, if you didn't see it, a 0. If you saw a few, it was 1. If it was prominent, it was a 2. And they also came up with the scoring system that would be used for the fibrosis scoring. So they had the 32 adult cases read by 9 pathologists for a total of 576 reads, 18 pediatric cases read by one time by the 9 pathologists for 162. So a lot less, I think, than what we're seeing these days in many of our clinical trials, our phase 2 studies certainly. And they agreed that a reasonable agreement between pathologists would be set at a kappa of about 0.5. One of the things that they did kind of focus on is the fact that the combination of these 3 criteria didn't necessarily indicate that the patient had NASH, but that these were just key elements that we could use to help track the progression of these findings over the course of time. So how well do we do in using the scoring system? So in 2005, when they first came up with the scoring system, remember their criteria was a kappa of greater than or equal to 0.5. Steatosis did pretty well at almost a 0.8. Ballooning made the criteria, the cutoff at 0.56. Inflammation at 0.45. They didn't do kappa on the NAS score. And fibrosis, they did a really nice job, 0.84. And here we go with that diagnosis, or the gestalt that I've heard many of our pathologists use in describing their pathology review, the diagnosis of NASH at 0.61. About 15 years later, and many of us having used this in a number of clinical trials, they decided to repeat the experiment and see how well we've evolved in being able to utilize this scoring system. Have the years really taught us anything? And you can see that not really. 15 years later, our steatosis kappa scores were about the same, ballooning about the same, inflammation about the same. We came up with a kappa score of 0.52 at this point for the total NAS score. Fibrosis came down a little bit to a 0.75, and the diagnosis of NASH about the same, 0.66. So 15 years of using the scoring, and we, in general, we're probably not much better than we were when we first started. So a couple of different studies have been done. This is just one of them that looks at our intra and inter-observer variability. In this particular study in 2011, they decided to take 2 pathologists, do some intense training with them, and see how we would could potentially improve on their pre and post training inter and intra-observer variability. And you can see that in general, that intra-observer variability did somewhat improve in almost all of the different scores that were there: steatosis, fibrosis, inflammation, as well as the ballooning. When we went to the inter-observer variability, we looked at the pre and the post, we actually kind of didn't do quite as well. And I think what was happening here is that they were so intent in trying to make sure that they were trying to be consistent with their colleague that perhaps they second-guessed themselves a little bit. And this is probably the key challenge that we faced over the years in being able to utilize the scoring system, not only for that eligibility criteria, but then also at the end being able to determine whether or not there's truly been a change in the treated versus the untreated patients in our clinical trials. We looked at, we've been talking about kappa scores, and for the statisticians, that means a lot. But for some of us, maybe looking at percentages may kind of bring home the message a little bit more. So in this study, they used 3 pathologists. They scored steatosis, a number of biopsies, steatosis, inflammation, and ballooning. And when you look at the steatosis, we went from about 22% to 29% of the cases where there was disagreement between the 3 pathologists. If you looked at the inflammation, we were from about 40 to 43. So again, quite a bit of discordance there. And then when we got to the ballooning, even more so, we went from 22 to 47% of the cases showing a lot of discordance. And if you've worked in these studies and you face the challenges of including patients, you're often faced with the fact that the ballooning is the one criteria that many of our patients didn't get that score. As you know, we often tried to set up an eligibility criteria where each of the different elements, steatosis, inflammation, and ballooning, have to have a score of at least 1, meaning that there's some level of abnormality, probably a score of 4, showing that there's a little bit more than just a little bit of disease, so that hopefully by the end of the trial, we can see some change with that. And meeting the criteria for ballooning has been one of the more challenging ones. And as you see from the results for the steatosis is usually pretty good at being able to capture that. Around the same time, as we move to the next slide, I believe we were at the NAShtag meeting in January with Dr. Harrison, who presented the results from the Eminence study. And one of the things that came up at that point was the fact that as they were using a reading paradigm where patients or slides were read in the beginning to determine eligibility, and then they were reread at the end to try to look at change over time, looking at them at the same time so that we could try to eliminate the concept of temporal drift on that. About 25% of the patients, when they were reread, didn't meet the eligibility criteria. And then even the diagnosis of NASH, about 13% of them didn't even meet the diagnosis of NASH when they were reread at the end. And so this kind of started, I think, in my mind, the challenges of the read paradigms and trying to come up with a better scenario where we could minimize that variability and try to make sure that we properly identify the patients at the beginning for eligibility, and then also be able to optimize our ability to show change over time. So where have we evolved over time? So we started out with some of the logistical challenges of glass slides. As you can imagine, glass slides, not very easy to kind of move around. So it often became a single reader paradigm. As we realized that the variability was there, we tried to implement a multi-reader paradigm, but the concern of losing the slides, of a breakage of the slides, and also the turnaround time, being able to move these across different regions where we had these expert pathologists to evaluate them, added to that turnaround time. As technology kind of caught up with us, we had the capability of creating digital slides. Again, they started out with that single reader paradigm and then decided that we were going to move to a multi-reader paradigm. And so in the imaging world, that 2+1, where you have 2 independent pathologists reading these slides, and then if there's a discordance between the scores, it then goes to an adjudicator. And even in that scenario, there were kind of a lot of different options that we were able to use. In some studies, sponsors and KOLs thought that it made sense to have only those elements where there was discordance. So if reader 1 and reader 2 disagreed on steatosis, the adjudicator only got to pick they agreed with reader 1 or they agreed with reader 2. In some other studies, it was felt that we should have that third person be completely independent. And so what you ended up in some cases was 3 separate scores, a 0, a 1, 2, and a 3, or a 1, 2, and a 3. So it really didn't help in terms of trying to get us to a consensus on that scoring. One of the other things that I think came into play during all this was something that Rob alluded to at the end, and that was, the slides themselves and what type of tissue we present to them. So in order to try to improve on turnaround time, initially, many of the studies had us using stained slides from the sites. So if you imagine that there were 20, 30, 40 sites maybe being used in these trials, you had 40 different pathology centers creating their own stains, their own cutting of those paraffin blocks. And so that may have added to some of the variability and some of the challenges. As we got to the point of the digital slides and we had to get them to one place where we could standardize that digitization, it also made sense that we would standardize who was going to do the staining from them. And so in most of our studies now, if not all of them, we're looking for at least the unstained slides so that that central lab, that histopathology lab, that one like MRL here at Medpace, can standardize and ensure that over time, there's no difference in the way that these slides are stained, and that again should minimize some of the variability that we see. So where are we now? Currently, the last couple of studies that we've been doing, we've evolved to a 3-reader paradigm where all the slides are being read by 3 pathologists at the same time. Digital platform makes that possible. So if we move on to the next, how does that work? So it's easy. If the 3 pathologists all agree on the different components, that's great. We have perfect consensus, and that becomes the final score. In the scenario where 2 of the 3 agree, we at least have what's referred to as the mode. 2 of them have agreed, and we don't have to worry about so much about that third. If the 3 of them disagree, then we come up with the median score. So we've got a 0, 1, 2, and 3. We kind of take the average of that and round it to the nearest whole number. And all of this is trying to minimize some of that variability that we see, and also help speed up some of the turnaround time and not delay things for being able to get that patient enrolled into the study. Obviously, if the 3 pathologists completely disagree on the liver disease, we can still call a consensus panel. If 2 of the pathologists deem that the sample is unevaluable, then we will go ahead and request another set of slides and restaining of them, with the hope that we can start again and improve on that process. So this has worked out rather well at this point with a lot of our studies. One of the things that we start out with, though, is trying to ensure that we get a certain level of concordance up front. And so we use a set of sample slides that all of our pathologists will review, and then we meet with them to ensure that they get an opportunity to discuss those cases where they've disagreed on some of the different criteria, and we try to refine what they're doing and how they're looking at these different slides, so that hopefully during the course of the study we can increase our concordance across all of them. And that seems to be working rather well, and seems to be the latest and greatest variation on a central read paradigm that's used in these clinical trials. So I'm going to turn things over now to my colleague, Nicola, who will kind of summarize for you, and then we can get on to some questions. Thank you.
Thanks very much, Sal. It was very interesting to hear from everyone, and really just to try and summarize, I think we can agree that liver biopsy remains essential in MASH trials as things stand. It's very clear that they are required as per regulatory authorities to meet these endpoints to demonstrate, efficacy of the drug, these composite endpoints of steatohepatitis, resolution and improvement in fibrosis. So at the moment, liver biopsy is here to stay, although we do acknowledge that there is increasing use of non-invasive tests, but so far they have not demonstrated that sensitivity and specificity, although appear to have an increasing role in perhaps finding those subjects that might have eligible biopsies. So as a way of finding subjects that could go on to biopsy is something to consider. We've heard obviously that it's really important to try and reduce variability in the biopsy handling, so collection, handling and processing, and how important that is to standardize the process. And also now we've heard that there has been a huge advancement in how the images are obtained. So we're looking not moving from the glass slides to digital images, and also optimization of the reads of the histology slides. So as Sal outlined, there's been this evolution really of this central read paradigm to now this 3-read pathologist approach. And I suspect over time, no doubt, that may evolve even further with time. On that note, I will now hand back to Corey so that we can take some of your questions. Thank you very much.
Excellent. Thank you, Nicola, and thank you everyone for your presentation. Very insightful, great content, and we are now ready to begin our Q&A portion of the event. As we do so, I invite the audience to continue sending your questions or comments. Just a reminder that if you're attending via GoToWebinar, please use the questions chat box. And for those of you watching on LinkedIn Live events, feel free to post your questions using the comments tab. We have already received quite a few questions, so I'm going to go ahead and kick off with the first one here. And rest assured, continue sending your questions in, and if we can't get to it in the time we have left, we'll have a chance to follow up after the webinar. So first question that's come in here, probably best suited for Rob to take this one. We've talked a lot about, obviously, liver biopsy here today. This audience member was just wondering, for accurate or the most accurate diagnosis, is there a preference on the segment of the liver that is most suitable for biopsy?
Yeah, actually, that's a good question, and it's alluded to or mentioned in the paper that I have referenced there in the slides. So among the group that co-authored that paper, it was felt that the biopsy from the right lobe of the liver was most appropriate because it was most easily accessible percutaneously. And then histologically, if one biopsies from the left liver, the fibrous capsule of the left lobe does introduce a little bit more a look of fibrosis, which can bias or influence the read by the pathologist. So the right lobe of the liver is the preferred lobe for biopsy. And in that same publication, I'll also mention that if possible, the same operator should be the person who does both the screening biopsy and end of study biopsy if possible, and also to obtain that biopsy as close as possible to the initial biopsy site. So you're really kind of comparing near adjacent or neighboring portions of the liver tissue.
Great, thank you so much for that, Rob. And Rob, I'm going to stick with you actually for a moment here because I think the next question probably pertains to you as well. This audience member was asking about how the unstained tissue section from the historic block, how exactly is that stored? Are there temperature conditions? How long would it still be useful for a stain?
So the stained slides can be, the unstained tissue sections can be stored ambient, room temperature. But let me add this caveat. So in terms of a clinical trial, in the context of a clinical trial, the preference is to have freshly prepared, uncut, unstained tissue sections sent to the lab for the staining. So in other words, do not cut the paraffin block up front and store those unstained tissue sections for a prolonged period of time. Now that's not, we haven't really determined what that time period is, but those unstained slides are exposed to oxygen, to air, to moisture. And so the staining quality will decrease over time. Now that being said, for a clinical trial, we like to receive freshly cut slides, which means that we will want to stain them within 30 days of the cut date in order to produce the best quality stain for the pathologist. Now, if the site is communicating with the local path lab, that's great. And if they're going to pre-cut those sections up front, we still want to receive them in 30 days of the cut date at that local path lab. So we're staining the slides within that 30-day period.
Great, excellent. Thank you so much for taking those two questions there, Rob. Another question that's come in here from the audience, and I'll leave this one open to whoever feels like they could field it. This audience member is asking about, so with whole slide digital imaging systems now being utilized, what, if any, technological issues have emerged?
Thanks, Corey. This is Rob. I'll take that one too. So yes, there have been some issues that we've encountered with connecting these globally located pathologists to the central histology lab's whole slide digital imaging system so that they can review and score the liver biopsies. And I would put these into basically three categories, if you will. The first category or issue we've encountered is the device incompatibility. Some of these pathologists use PCs, some of them use Apple Mac products. So the type of whole slide digital imaging system that's used by the central lab may not be compatible with a Mac or Apple product. And that's something that can be worked around by just getting a PC instead of using a Mac. Also with Mac, Apple products, sometimes the virtual private network, the VPN, is incompatible with an Apple product. So obviously we want to have a VPN connection between the pathologist and the central lab in order to ensure a secure communication between the two. The second category, I kind of put it into like institutional firewalls, antivirus, anti-malware programs that may be out there. And sometimes whatever the academic institution or company have installed on their laptops, their computers, may also be incompatible with the central histology lab's security, whatever security system they have on their system. And then I think the third one that I'll put in there is the VPN requirements. Whatever central histology lab is doing the work, that central lab's VPN will need to be installed on the pathologist's personal or workplace computer, just to ensure that there are secure communications between them. And then here we kind of run into issues because it involves getting the institutional IT department involved, which will have system rights to permit the loading of that VPN program onto their computers. That takes time. And then obviously there's the question of whether a central laboratory would want to even get involved with installing a VPN onto a personal laptop in case something goes wrong and the pathologist's personal computer crashes after the installation. So that's another issue to address as well. So we've got kind of like three categories that I've said there. All of those do take time to resolve and can delay the study setup from the histology lab's point of view. They're not insurmountable, but they do delay somewhat the getting the pathologist up and ready to actually do the reads on the digital images.
Excellent. Thank you again, Rob. Another question here from the audience. Hopefully we can give Rob a break on this one. We'll see if any of our other panelists, this fits your area of expertise. This audience member was wondering about how you envision read paradigms evolving in the future.
Okay, maybe I can take this one and give Rob a break. This is Sal Zavatino. So, we've seen a lot of these read paradigms shifting over time, number of pathologists. What we keep seeing is that the disagreement between pathologists often ends up being in those cases where it's borderline. You're going from a 0 to a 1, a 1 to a 2, or a 2 to a 3. And as we heard earlier on, there is a push now in many of our computer-based systems to have AI assist us in being able to do things. And so what I see, or why I envision, is that some of these AI tools that are being developed out there, once they get approved by the regulatory agencies, that they can be utilized to help assist the pathologist, especially in those cases where they are sort of borderline between those different scoring systems. I don't think that the AIs will completely replace the pathologist, because there's still a lot of value in terms of other diagnostic criteria that might come up as they're reviewing these different patients. But certainly for the purpose of scoring, helping during those cases where you're sort of on the fence, it can add as a tool for them and help minimize, I think, some of the variability. It'll be nice to see how that works out in the next year to two.
Great, thank you so much, Sal, for your answer on that one. Another question that's come in from the audience. I think we have time to hit a few more questions here. One that I was actually wondering myself through your presentation, this audience member was asking, how soon should we actually be repeating biopsies in studies?
Thanks very much, Corey. I'll take this one. Yeah, it's a very good question. I think obviously we want to try and minimize the number of biopsies that are performed in a study due to obviously the impact of on enrollment. I think subjects will not be very happy with having multiple biopsies throughout a study, and also the associated risks with each procedure, as we heard from Jensen earlier. You want to allow enough time really to observe a change in histology. There are components that can obviously change more quickly than others. We know that steatosis can improve more quickly, sort of over weeks rather than longer time frames, but other histological changes such as fibrosis take a lot longer. I would say that currently most liver biopsies are performed at least a year after the initiation of treatment. And it's really to ensure that you're observing a maximal histological response, really to make sure that you're hitting those clinical endpoints. So you want to see the maximal effects of the drug on liver histology.
Great, thank you so much, Nicola, for clarifying that and for expanding on it. Another question here from the audience that's asking about if there's any potential variability to consider between the different equipment available. I mean, we've talked a lot about different equipment today. How does variability factor into that?
Well, this is Rob. I can address that one. So if we're talking about the instrumentation within the histology lab, yes, there will be some variability. So for example, most labs have an automated H&E stainer, and they will use an IVD approved kit that's appropriate for that particular machine. Now, that being said, there are differences in the hues and colorations of the stains. But again, those kits are IVD approved, so it's just it's a pathologist just having to adjust their eye in terms of the read and looking at the hues and colors between the pinks and the blues or whatever colors they're going to use or whatever histologic features they're looking for. There will be those differences, but again, I think most pathologists can look through those differences in terms of an H&E. When it comes to a trichrome, which is often used for assessing the fibrosis of these biopsies, some labs do a manual hand dip trichrome stain, which will induce a much more variability because it's manually done, and those solutions have to be made up each and every time. Some histology labs do have automated special stainers, again with a dedicated trichrome stain. Those would be preferable. And again, there are different manufacturers out there, and again, the coloration may differ between the machines, the platforms themselves, but again, a pathologist should be able to read through those as well.
Well, thanks very much for that, Rob. And I think we have time for a couple more questions here. And you just sort of ended your previous question talking about, pathologists having time to read that. Great question that's come in here is asking, when would a reread be recommended in a study?
Maybe I can take this one and team can add to it. So I think the fact that we now have a read paradigm that's utilizing 3 readers on all cases already gives us the inter-reader variability and helps minimize some of the variability that you might see in an individual score. So the need to reread, I think, is minimized from that. I think the rare cases where you might want to reread something might be where unfortunately, the pathologists that started out in the study are no longer available for one reason or another, and that team of pathologists has now changed during the course of the study. And so working with our sponsors, working with our statisticians to try to see what the magnitude of that on the overall score might be could help guide us on whether or not we do need to do a certain amount of rereading on that. But again, if you look back at the study that I referred to earlier on in our presentation, you do run into a potential where you now have discordance in what you included and what you didn't include in terms of patients with that. So I think you would move cautiously in that reread. But again, the fact that you've got 3 pathologists now reading all of these cases minimizes some of the concerns that we had up front with the intra-reader variability issues.
Great, thanks very much, Sal. Anyone else on the panel wanted to add to that? No, I think, okay. We'll move on then to our, I think will be our final question here today before we wrap up. And we haven't heard from Jensen yet during the Q&A, so I think Jensen, this one's probably up your alley. This audience member was just asking, we've talked a lot about liver biopsies today, but could the FIB4 index, could that actually be looked at as an alternative to a biopsy? Would that be a possibility?
Thanks, that's an interesting question. So FIB4, fibrosis 4, is a biomarker, so it's one of the non-invasive tests, the NITs that we look for detecting fibrosis, and this is based on age, AST, the platelets and ALT that the patients will elicit. So it is a very good indicator for fibrosis. The problem is it's not approved as an endpoint from a regulatory perspective and a marketing perspective. And just to compound further, retrospective studies in this have looked into it, and there are things that can inadequately place FIB4. So age is one of them. It's also impacted by AST. So if there's been exercise recently, alcohol recently as well, then the values you're going to get from your FIB4 are not going to adequately relate to the levels of fibrosis that the patients are actually showing. So it is a great indicator, and it's good to have it as a tool to identify which patients would you then put through to your biopsy, because it's rather inexpensive. You're already collecting their age and the other biomarker and the other lab assessments. So it's very helpful to implement FIB4 and those other non-invasive testings to determine who should be going and progressing to the biopsy. So it should be used in combination.
Great, thank you very much for that, Jensen. Really interesting and fascinating, and I think a great point for us to end here. I do thank yourself for that answer and all of our speakers for not only the answers during our Q&A session, but for the great presentation. A reminder to our audience, if we couldn't attend to your questions, or if you have any further questions, or if you're someone who's watching the recording of this webinar, you can get in touch with the team at Medpace using the contact info up on screen. There's just the email address there, info@medpace.com, and they'll have a chance to follow up with you after today's presentation. I do thank everyone once again for participating today. If you are attending via GoToWebinar, I've added a link in the chat box where you'll be able to view the recording of this event, and you can of course share this link with your colleagues so that they may register to view it as well. You will also be receiving a follow-up email from XTalks that has access to this recording, and after we wrap things up here, a survey window will appear on your screen. Your participation in the survey is always appreciated as it helps us to improve our webinars. For those of you who are watching on LinkedIn Live, we do encourage you to create an account on xtalks.com and register for this event, which will then of course give you access to today's recording, but also our entire library of webinar content. Now, please join me in thanking our speakers once more for their excellent presentation. Sal, Rob, Nicola, Jensen, thank you so much for your time. We hope the audience found it informative, and we wish you all a great rest of your day. Take care, everyone.